So this is my introductory lecture to our laboratory exercise on biochemistry. And this week we're going to be running 4 different tests. We'll be learning about 4 different techniques that are used to detect the presence of 4 of the kinds of molecules that we've talked about in lecture in Module 3. Okay. Polysaccharides are the first molecule that we'll be talking about. We think of them as either starch or glycogen. And there's a really easy test that we use to detect the presence of polysaccharides, which involves regular old iodine. It's actually iodine potassium iodide, and it's the stuff that maybe some of you have used to disinfect wounds. So there's this, if you've ever had a little owie, a little raspberry on your elbow because you're sliding into second base and you got scratched up, maybe somebody applied some tincture of iodine, which is kind of a brownish material that would have turned your skin kind of brownish in the area around the wound. The general idea there is that this material is kind of toxic to bacteria, so you're less likely to get an infection, but it still stains. I mean, I think probably nowadays very few people are going to be using a medicine that will stain a kid's skin dark, when there are other kinds of medicines that don't do that, but I think that there's still some parents that still have some of the iodine stuff around. Okay, the basic idea here is we've got this material. It's called IKI for iodine potassium iodide. This is material that's there in both tincture of iodine as well as Lugol's reagent, which is the stuff that we'll be using. Basically what happens, we're taking advantage of the fact that polysaccharides, like starch, are made up of, remember this is a long chain of individual glucose molecules, but it's not just a chain. This chain is kind of set up in a little coil structure. We don't have simply a chain like this where all the glucose are hooked up in a linear chain. That chain actually kind of twists on itself, and we get this kind of coil structure, which Lugol's reagent, the IKI, this molecule of IKI is going to be fitting in there, which causes a change in the structure. It makes what's otherwise a transparent or not terribly visible thing, it'll turn this dark blue. It's a really nice and easy thing to read. We'll be using this IKI with starch reaction in next week's laboratory to detect the presence of starch again as we're doing our enzymes lab. So in any case, that's our system number 1. Polysaccharides, by which we mean things like starch and glycogen and even amyloids. Okay, maybe some of you have heard of amyloids, which is a condition that we associate with lots of diseases. In particular, people are concerned about Alzheimer's diseases, which is associated with the building up of amyloid plaques around the neurons. So basically what I'm telling you here is that amyloids are called amyloids because they're similar to these polysaccharides. And one of the ways that they're similar is that they tend to stain similarly to polysaccharides. In other words, even though they're not really made up of chains of glucoses, they'll react with reagents, sometimes even Lugol's reagent, and it'll seem like there are these starches in place when they're actually these protein-based amyloids. That's a little aside for you guys. And the second kind of molecule that we'll be looking for and detecting are molecules of simple sugars. Things like glucose and sucrose and maltose and lactose. In particular, these are sometimes called reducing sugars, which is kind of like a chemical way of saying that these are the sugars that we care about. These are the ones that tend to be sweet, not just glucose and fructose and sucrose, maltose, lactose. Basically, all the sugars that we associate with food have this tendency to have to reduce things. Reducing things means that you are putting additional electrons on a molecule. So it's kind of weird. You think reducing is taking something off. No, reducing is actually putting electrons onto something. And an examples of this again would be things like glucose, fructose, which are the sweet sugars that we have in foods, and sucrose, or table sugar, is simply just a glucose and a fructose. Remember, glucose plus fructose is equal to sucrose. Sucrose is an example of a disaccharide. The thing about these reducing sugars is that they will have this reducing reaction. They'll react with an alkali to create this thing called an enediol, which then will interact with copper to form a precipitate that is basically easily visible. So we have copper that is in solution. Yeah, that's right. You need copper. We think of it as a metal, but the individual ions of copper would go into solution very nicely. And if you have these sugars interacting with an alkali in the presence of the copper ions, what happens is you get this precipitate. It's very easy to detect in a solution. Okay? I know I made it sound really complex, but it's really kind of easy. You start off with a test tube, and the test tube is going to have some the material that you're testing in it, right? So here's the material. I want to ask the question: is this material containing reducing sugar or not? And so I'm going to add another quantity of the Benedict's reagent, which is blue in color. And after we mix it all together, the whole thing is going to be blue. And now what we need to do is to apply heat. And after heating this thing up, the solution might turn color. And basically the color that it turns is how we tell how much sugar there is in the solution. So if it were to turn brick red, then that would mean that you would have a really strong reaction. This would be a really strong positive for the presence of a reducing sugar like glucose. Okay. If you have a weaker reaction, it might not be quite as red. It might be kind of ranging from orange to yellow and basically to green. And maybe a very weak green result would indicate some positive reaction, but a relatively weak one. Okay? So the test that we use for simple sugars is going to be the Benedict's reagent. The test that we use for polysaccharides is going to be the Lugol's reagent. It's going to be based on this Lugol's reaction. With the case of Lugol's reaction, a positive is going to be a dark blue color, whereas in the case of the Benedict's reagent, this really strong positive result is going to be any kind of color change. A negative reaction would be one where it doesn't turn color much at all. It might just stay the bluish color. So the next kind of test that we want to focus on is going to be a test for protein. And you should remember that a protein is the same thing. Basically, the basic structure of a protein is a polypeptide. And a polypeptide is a long string of amino acids. And the reagent that we're using here is called Biuret reagent, which is actually kind of similar in nature to the Benedict's reagent. In fact, you might find it a little bit confusing that this Biuret reagent looks almost the same color. It's a little bit lighter. It's almost the same color as the Benedict's reagent. You don't want to confuse them. A Biuret reagent, by the way, is very, very basic. It's got a high pH. It actually has a pretty high concentration of sodium hydroxide, and it will cause a burn. It'll cause a burn if you were to get it on your skin. So I guess safety issue number one, safety concern number one. If you do get Biuret reagent or any kind of alkaline material on you, you could rinse it with water. That would be good, but it's also a good idea for you to apply something that would neutralize a high pH challenge or insult to your skin. So if you have a high pH insult to your skin, the appropriate thing to wash with would be like a dilute vinegar solution. Vinegar is acidic, but it's not so acidic that it would cause you to be burned. You know, obviously we could neutralize Biuret reagent with hydrochloric acid, but that would be a really bad idea for you to wash yourself with hydrochloric acid. By washing with vinegar, you're both neutralizing the strong base that's there in the Biuret reagent, and at the same time you're not really creating any further problems for yourself by applying a really strong acid. Okay, so that's our little safety note. So Biuret reagent is this blue stuff. Similar to what we did before, you would add it to a substance. If your substance is here, that's your question mark. You would add a quantity of Biuret reagent. You would mix it all up so it's going to uniform in color, and at that point, it'll either turn color or it won't. And the reaction that occurs here is actually a reaction that involves a peptide bond, which is the bond that occurs between amino acids. So you might remember from lecture that if one amino acid kind of looks like this, where you've got an amino group on this side, and we've got a carbon in the middle, and we've got an R group, that's the R group over there, here's the hydrogen, and the third carbon is going to come over here. There's a bond between this amino acid, this is one amino acid, and the next amino acid, which is going to have an amino group, a central carbon, and another carboxyl group. Okay. And it's this bond right here, right there, that will interact with the copper and with the high pH. Basically, this is going to be changing its distribution of electrons, which is going to cause the blue color of this Biuret reagent to shift to kind of like a lavender color. Okay, so the lavender color, if you think about it, it's actually kind of in the opposite direction relative to what we saw in the case of the Biuret reagent. One little side note here is that the Biuret reagent and the Benedict's reagent really do contain pretty much the same reacting compounds. It's still copper. It's the same copper reaction. So if there is any sugar in the material that you're testing using Biuret reagent, it's entirely possible that you might be getting some kind of weak Benedict style reaction. So being really specific here, in the case of the protein, we're looking for a reaction, a color change that's going to take us from an original blue to kind of a lavender color. Any change towards green or any change towards yellow would basically be something other than the Biuret reagent color. In the case of Benedict's, this is a reaction that really does go through a sequence from green to yellow to orange to red to this dark brick red color as a gradation of increasing strength of reaction. So Biuret reagent, this is the one we're going to be associating with our test for the detection of protein. Last test we're going to do is going to be the Sudan III test, which is going to be the test for lipids. And remember, lipids are greasy, waxy, oily molecules because of their highly nonpolar nature. They're hydrophobic. And things that are strongly hydrophobic are going to be associating with lipids. Things that are strongly hydrophilic are going to tend to repel lipids. So basically we're taking advantage of that general premise that we talk about in BIO100, which is like dissolves like. So if you're a lipid, you're going to interact really strongly with other things that are also very lipidy and very hydrophobic. And we're going to be taking advantage of this. We'll be using a stain called Sudan III, which is a lipophilic stain. It's actually, this is actually the dye that's used to colorize fuels. If you've ever poured regular old gasoline, the stuff in your tank, into a clear container, you might have noticed that it's kind of an orangish color. And that's kind of misleading because you might think that gasoline is orange by nature. No, gasoline, the material that is the fuel for your engine, is really a clear liquid. Sudan III is used to stain clear liquids so that we're able to recognize those fuels like gasoline and diesel fuel, and things that are going to be taxed have to be distinguished from other kinds of fuels that are not subject to the same degree of taxes. So Sudan III is actually a pretty widely used dye, and it will allow us to detect the presence of lipids. And here we're going to be taking advantage of the fact that regular old filter paper is really quite hydrophilic. It's made up of cellulose, which is made up of glucose, and you already know that carbohydrates are relatively hydrophilic. So paper on its own doesn't have a very strong tendency to interact with lipids, but the structure of cellulose, the structure of like a paper towel, the fibrous nature allows it to soak up lipids to a certain degree. You know this because when you spill some oil on a table, you can use a paper towel to sop it up. I mean, the fibrous nature of the paper will allow it to absorb some lipids, even though it doesn't necessarily like to interact with lipids. So if you had some paper, let's pretend this is paper here, nice and white. You might be able to blotch some lipidy material here. Maybe it's grease from that carne asada burrito you had for lunch. So if you were to have like a paper bag and you had a nice greasy blotch there, this is all greasy stuff in here. You might let it dry out. You wouldn't necessarily see it as being a different color. There's no necessary color there. But by using Sudan III, we could start by dropping this whole piece of paper into the Sudan III, in which case we would get pretty much the entire world becoming kind of this color. But the paper is going to have a tendency to decolorize relatively rapidly. If we take that Sudan III and dip it in water, and let the water kind of carry the Sudan III away, or let the paper start interacting with the water like it really does, which would cause the Sudan III to go away. The paper itself would kind of decolorize, but this area here where you actually had the lipidy stain, it's going to retain that color. Okay? So in other words, if you have a material that has got some hydrophobic substance in it, and you could apply it to paper, let it dry, and then use Sudan III to detect the presence of lipids in that sample you blotted onto the paper, and that's going to be the basis of the Sudan III test. So the way this lab works, you could read the rest for yourself actually, but I'll give you the quick rundown here, is that you're going to be running kind of a set of pilot tests so that you know what a positive reaction and a negative reaction looks like for all four of these tests. For the polysaccharides, we'll be running a whole bunch of tests. We're looking at regular old water. We'll be testing water for polysaccharides. This is going to be our negative control, right? We need to know what a negative reaction looks like, so in case we actually see a positive reaction, we'll be able to know it for sure. Then we'll actually have a known positive, known starch. And so if you actually have starch in the solution, you'll be able to see really clearly this kind of reaction that takes place. We also have a couple of other samples that we'll be testing. They will be testing glucose and honey, and no, maybe not honey. We'll be testing glucose and egg white. Egg white is there as kind of like our positive test for protein. For some reason, they decided that we should just test all four of these things: water, starch, egg white, and glucose. We should run all four of these things through the tests for polysaccharides. I guess the idea here is that you want to convince yourself that that starch really does give a positive reaction for this Lugol's reagent, whereas glucose, which is basically also made up of the same material as starch, except that it's monosaccharide as opposed to polysaccharide, glucose actually gives you a negative. Also, we're basically establishing that IKI does not have the same reaction with egg white, right? Okay, so it's like this. We're doing the water as a negative control, the starch is going to be a positive control, and glucose and egg white are there just to kind of convince ourselves that this polysaccharide reaction with Lugol's is selective. It's not something that will just interact with any old thing. It has to be the right thing. Okay, now we do the same thing with simple sugars. We'll be taking the Benedict's reagent and testing water, glucose, that's our positive control, and we're also going to be testing starch and egg white. Yeah, right. I mean, because obviously this is going to give us a negative result. This is going to be giving us a positive result. But you might say, well, starch is made up of glucose, and if glucose can reduce the copper ions in Benedict's reagent, why can't starch? Well, it turns out that the only parts of the starch that can reduce the copper ions are those molecules at the very ends of the chain, right? So in a polysaccharide, you've got a really, really long chain of glucoses, and it's only the molecules at the end that might have the opportunity to cause this reaction to take place. And we don't have very many of those ends, so there really isn't a strong Benedict's reagent reaction. In fact, you're not able to detect it at all. Egg white, similarly. For all we know, there might be something in egg white that also causes it to react with the Benedict's reagent, and we want to make sure that that's not going to be happening, so that we can interpret our results later on with great confidence. Okay, the same thing here. Protein, we have the Biuret reagent. We'll be testing water. Obviously, it's going to give us a negative result. The egg white, albumin protein, and egg white is definitely going to give us a positive reaction, so we'll see the purple color as opposed to the egg white. Then we'll have glucose and starch. Again, there to let us confirm for ourselves that we don't really have any reaction taking place with the Biuret if we've got simple sugars or polysaccharides. Same thing goes with lipids. We run all four of these materials. We run them on lipids as well, and we get a negative result in all of them. And for that reason, we have to bring in a fourth material. We'll be testing vegetable oil. We'll have a positive control vegetable oil, which will allow us to see for ourselves, see firsthand what a positive result with Sudan III is going to look like. Okay. And after all that, after we go through those first rounds of tests, so that we have a good comprehensive picture of what positive and negative results look like for these four tests, we'll be running these 4 tests on some unknown materials. And that's it for this introduction. I'll see you guys in the laboratory.